In this session, I'm going to deal with the histology of the digestive system. The gastrointestinal tract is essentially a tube, and this tube has four distinct functional layers. These are the mucosa, submucosa, muscularis propria, and adventitia. Some of them, like the mucosa, they have sublayers. Like the mucosa has the epithelium, lamina propria, smooth muscle layer, which is called the muscularis mucosae. And then the muscularis propria is subdivided into inner circular and outer longitudinal layer in most of the places. And then there is the adventitia. Now let's apply this on an actual section from the gastrointestinal tract. This is a section showing the esophagus. And you can see here that this region is the mucosa. It consists of epithelium. In this case, this epithelium is a stratified squamous epithelium, non-keratinized. And then there is a lamina propria of connective tissue. And you can see here that there is a muscularis layer. These are muscle fibers, smooth muscle fibers. They constitute what is called the muscularis mucosae, or the muscle layer of the mucosa. And then beneath the mucosa, the other layer is the submucosa. You can see here, again, it consists of connective tissue. It has some glands here, mucus secreting glands whose ducts will pass through the mucosa and open on the surface. And also there is a plexus of nerves, autonomic nerves. And then we have the third layer, is the muscularis propria. They call it propria to differentiate it from the muscularis mucosa. And this muscularis propria has two distinct layers. You can see the muscle fibers here are in two different orientations. Here, this is one orientation, and this is the other orientation. Here, the orientation of the muscle fibers is like circular, and these are longitudinal muscle fibers. You can see that the muscle fibers are cut across their longitudinal axis. Between the two layers of muscle fibers, you can see here traces of nerves, nerve plexuses, another nerve plexus, autonomic nerve plexus. And then we have the adventitia, which is formed of connective tissue, contains blood vessels, nerves, but not a plexus, but not an autonomic plexus with ganglia. And in some places, it is covered with mesothelium, which is the peritoneum, the visceral layer of the peritoneum. This is again here another section from another part of the gut tube. It is from the small intestine, and you can see the villi here. Although the features might look different, but essentially they are the same layer. The villi are covered by epithelium, and there's a core of lamina propria in the villus. And then you can see the muscularis mucosa here. So these three layers together will constitute the mucosa. And then we have a submucosa, which in certain parts of the small intestine, it contains submucosal glands, like in the duodenum, Brunner's glands, and then the muscularis propria with inner circular, outer longitudinal, and then there is the adventitia. So essentially the same four layers. In the gut, there is lymphatic tissue, and this will provide a defense mechanism because of the continuity of the gut with the external environment. And this, what we call gut-associated lymphoid tissue, or GALT, is distributed throughout the gastrointestinal tract. It might be diffuse, so you can see diffuse lymphocytes, diffusely scattered cells, or it could be dense aggregates forming what we call lymphoid follicles. Like the example here, you can see the lymphoid follicles and the wall of the small intestine. Look at the villi here. So this is the small intestine. Also, they are present in the wall of the large intestine. An example here in this section, this is a section through the appendix, and you can see here the aggregates of lymphoid tissue. The autonomic plexuses in the wall, there are two places where autonomic plexuses are located. I mentioned that in the submucosa, there is what we call the submucosal plexus. And then in the muscularis, between the two layers of the muscularis propria, there's the myenteric plexus or Urbach plexus. These plexuses, they contain postganglionic sympathetic fibers, and they contain ganglia, but the ganglia belong to the parasympathetic nervous system, because the sympathetic fibers, they have already synapsed in prevertebral ganglia that are related to the branches of the abdominal aorta. So the ganglia here in both these plexuses are parasympathetic ganglia. Regarding the mucosa, there are 4 distinct types of mucosa, and these usually they change abruptly from one region to another. The mucosa can be either protective, secretory, absorptive, or absorptive and protective at the same time. And as I said, they change abruptly. So here at the gastroesophageal junction, the mucosa changes from a protective mucosa, which is this stratified squamous epithelium, non-keratinized, in the esophagus. It changes to secretory mucosa. These are the pits of the gastric glands of the stomach. And then here at the pyloric sphincter, what we call the gastroduodenal junction, the mucosa changes from the secretory mucosa of the stomach to the absorptive mucosa of the small intestine. You can see the villi here, and the mucosa, these villi, which are finger-like projections to increase the surface area. And also there are glands, the crypts, that dip into the mucosa, and sometimes they reach the submucosa, as you can see it here in the duodenum, the Brunner's glands of the duodenum. And in fact, as you can see here, the submucosa of the remaining part of the small intestine does not contain these Brunner's glands, but the Brunner's glands are a feature of the duodenum, will help you identify and differentiate the duodenum from the remaining part of the small intestine. And then we have the other place where the mucosa changes abruptly. It is here at the ileocecal junction. And in this case, the mucosa will change from the absorptive mucosa with villi of the small intestine, of the terminal ileum in this case, into the absorptive protective mucosa of the large intestine. You can see here that there are no villi, only there are simple tubular glands. And you also note that there are a lot of these cells that look like as if they are empty. These are called goblet cells. In fact, they contain mucus, and this mucus will be washed during the preparation of the slide, so most of the cell will appear as if it is empty. There is a lot of goblet cells if you compare this with the amount of goblet cells that are present in the small intestine. And this is for protection, because these goblet cells, they secrete mucus that will reduce the friction between the formed feces that will become thicker in the large intestine as most of the water is being absorbed. And then the fourth change in the mucosa is at the anorectal junction. The mucosa will change from absorptive protective to a purely protective mucosa. Remember that we had a protective mucosa in the esophagus, which is a stratified squamous epithelium non-keratinized. And so here at the anorectal junction, the mucosa changes from this absorptive protective mucosa to stratified squamous epithelium, which is a purely protective mucosa. I will show that to you in an upcoming slide. Let's start with the esophagus. So in the esophagus, we have the epithelium, which is a protective stratified squamous non-keratinized epithelium, and there is a lamina propria with diffuse lymphoid cells, scattered lymphoid cells, not follicles. And then we have the muscularis propria. These 3 layers, they constitute the mucosa. And then we have the submucosa with the submucosal glands, which are serous and mucous glands. You can see here the acini, but these are ducts that are lined with the cuboidal epithelium. The secretions, they pass through the ducts and open on the surface of the esophagus, on the lining of the esophagus. They aid lubrication, and they are most prominent in the upper part of the esophagus. Of course, these submucosal glands are present in the surrounding connective tissue of the submucosa. And then we have the muscularis propria with the inner circular, outer longitudinal muscle fibers. In between them, as I said, that there is the myenteric plexus. Remember here that the submucosa also contains the submucosal plexus of nerves. The presence of these 2 layers, the circular and longitudinal muscle fibers, will help in the peristalsis, so that food passes through the esophagus because of the peristaltic action of its muscles. This is aided but not dependent on gravity. So you can drink or eat even if you are in an upside down position or if there is loss of gravity as in space. To keep in mind that these muscle fibers and the muscularis of the esophagus and the upper part of the esophagus, upper third of the esophagus, they are skeletal muscle fibers, but they are not under voluntary control. And in the lower part of the esophagus, they are completely smooth muscle fibers, as elsewhere in the GI tract. And in the middle third, there is a combination of smooth muscle and cardiac muscle fibers. This is to show you the gastroesophageal junction, where there is an abrupt change of epithelium. And by the way, this is not a conventional stain, and that's why you see these colors that you might not be accustomed to, but the features are clear. You can see here that there are multiple layers of cells, so this is a stratified squamous epithelium. The superficial cells are flattened, and then at one point here, the cells will become columnar cells. And these columnar cells, they have a lot of mucus. The mucus takes the purple coloration. That's why you see a lot of these purple colored cells. So this is the stomach, and these are the gastric glands, the tubular glands here. These are the gastric pits leading to the gastric glands. This is the region of the muscularis mucosa. Here's the submucosa, and then the muscle layer. You can see that the muscle layer is not thickened. It's the same. Muscularis propria is the same. So there's actually the sphincter here between the esophagus and the stomach. The gastroesophageal junction is a physiological sphincter rather than an anatomical sphincter. An anatomical sphincter, like the one that is located at the gastroduodenal junction, means that there is a thickening of the smooth muscle fibers at the muscularis propria. In the stomach, the muscularis propria is formed of 3 layers. Apart from the outer longitudinal and the middle circular layer, there is an innermost layer which contains oblique muscle fibers, and having 3 layers here indicates that the importance of the stomach in physical digestion of the food, the churning action of the stomach. These are the gastric glands, and you can see the epithelium is a secretory epithelium. These cells are simple columnar epithelial cells. They are mucus cells, and they have a lot of mucus inside them. As you can see, the cytoplasm is pale because there is a lot of mucus, and the surface is forming these pits, gastric pits, and into the gastric pits, if you look at this diagram here, open these tubular glands. The cells here, there are different types of cells. I can see the mucus cells, as I mentioned. They are present on the surface, they are present in the pit, as well as in the neck of the gland. And the other type of cells are the parietal or oxynetic cells. And then we have the chief cells, and then we have neuroendocrine and stem cells. Now let's deal with these cells on separate basis. So the mucus cells, as I said, that they are filled with mucus. They are simple columnar epithelial cells forming the gastric pit, and also they extend into the neck of the gastric glands. This mucus is important to protect the epithelium from autodigestion. So it will form a layer of mucus. The mucus contains the protective bicarbonate ions in its deep layer to neutralize the acidity and autodigestion by the gastric juice. This is again the special stain that we used, and it will show you these cells, the mucus cells. They are purple in color because the mucus is stained purple, and you can see the location. They are on the surface, they are in the pits, and some of them are located in the neck of the gland as well, neck of the gastric glands here again. And they are squeezed between the parietal cells. These large cells here with centrally located nucleus, these are the parietal cells. So the next type of cells are the parietal cells, and they are present along the length of the gland, but tend to be most numerous in the upper part of the gland. As you can see here, this is the upper part of the gland. You can see it here in the eosine hematoxylin preparation. They are large cells, rounded. They have extensive eosinophilic cytoplasm with a centrally located nucleus, and usually they are described as having what we call a fried egg appearance. They secrete the gastric acid, the HCl, as well as the intrinsic factor. And this intrinsic factor is important for the absorption of vitamin B12 from the terminal ileum. The other type of cells in the gastric glands are called the chief cells, or peptic or zymogenic cells, and they secrete pepsinogen that is converted into pepsin, which is important for the digestion of protein. And you can see that most of these chief cells are located in the lower part of the gastric glands. You can see them here in this stain. Most of them, they are occupying the lower part of the gland. Look at the cells in the upper part of the gastric gland. Many of them are with eosinophilic cytoplasm, but these cells, the chief cells, they have a basophilic cytoplasm. If you look at the basophilic cytoplasm here, granular cytoplasm, and it reflects their large content of ribosomes, which are important for the production of pepsinogen. The nucleus is basally located, it's not centrally located nucleus, and they are more columnar. The other types of cells that are present in the gastric glands are neuroendocrine cells, and these are part of the diffuse endocrine system. Usually they are toward the base of the gland, and they secrete local hormones like histamine and other hormones, and you cannot identify them in conventional stains. So for example, here in the pyloric region, these neuroendocrine cells are called G cells, and they secrete the hormone gastrin, and in this histological section, they are clearly seen because this is an immunohistochemical stain using an antibody to gastrin. So that's why they can be specifically identified. Gastrin goes into the bloodstream and promotes the secretion of pepsin and acid by the gastric glands, by the parietal cells and by the chief cells of the gastric glands. And also it enhances the gastric motility. The other type of cells are called the stem cells, and they are found mainly in the neck region of the gland. They don't have a special feature, but we can say that this is a stem cell if we can see a mitotic figure, because these cells are continuously dividing and replacing all the other types of cells in the gastric glands. Now, so far about the stomach, let's deal with the small intestine. This is the principal site of absorption. Most of the absorption takes place, and in order to increase the efficiency for this part of the gut in performing its function, there are 4 factors that combine together. First of all, the small intestine is long, and you might have known that from the gross anatomy, that the small intestine is about 6 meters in length. And then the mucosa and submucosa, they are thrown into circular folds. You can see these circular folds. These are called the plici circularis, or valvulae conniventis, or valves of Kerckring. You can see them grossly when you open the small intestine, especially in the duodenum and the jejunum, and then they are going to fade gradually as you reach the ileum. In a histological section, you can see these folds. They are folds of mucosa and submucosa. Don't confuse them with the villi. These are the villi on top of them, and the villi are folds of the mucosa, but the circular folds are folds of mucosa and submucosa. These are the villi. This is the third factor that increases the surface area for absorption. So these are villi. They are folds of mucosa. You can see here they have a core of lamina propria, and they are covered by epithelium, simple columnar epithelium. On their luminal surface, they are characterized by multiple finger-like projections. These are called microvilli. So differentiate between the villi and microvilli. The villi can be seen clearly in such a slide, of light microscopic slide, but the microvilli are difficult to be seen. In the light microscopy, there is a hazy area which is called the brush border, because they look like a brush as seen by electron microscopy. Look at the brush border here, the luminal surface. It looks like a brush. That's why they are called brush border. They are the microvilli. And as an indication of the importance of these structures in absorption, loss of the normal intestinal villi with their, of course, microvilli will result in malabsorption, like in celiac disease, for example. In the small intestine, in addition to the villi and in between them, there are simple tubular glands. These are called the intestinal glands, or the crypts of Lieberkühn. And in a section like this one, it might be difficult sometimes to tell which one is the villus, which one is the gland or the crypt. So if you find that there is a lumen in the center of the profile, then it is a gland. But for example, here in this circular profile, there is no lumen. This is the
lamina propria,
but no lumen. So this is a section in a villus, but this one is a section in a gland. In the duodenum, apart from the villi, these are the villi, the plici circularis and the glands, Some of these glands, they dip into the submucosa and they form what we call submucosal glands or Brunner's glands. Beneath the submucosa, this is the muscularis, and so we are here in the submucosa, and these are the submucosal glands or Brunner's glands. They are exclusive for the duodenum and will help you differentiate the duodenum from other parts of the small intestine. These Brunner's glands, they are important because they secrete an alkaline mucus which help neutralize the acidity of the chyme that has just arrived from the stomach, and so it is important for protection of the duodenal mucosa from autodigestion. That's why they are not present distal to the duodenum. This chyme that reaches the small intestine will also stimulate the release of hormones from neuroendocrine cells. These hormones of secretin, cholecystokinin, and pancreozymin that affect the function of the pancreas and the gallbladder. These are the histological features of the remaining part of the small intestine. So I would say that this slide belongs to the small intestine because of the presence of the villi. So it's a small intestine. Which part of the small intestine? I look at the submucosa. I don't see any submucosal glands, so I exclude the duodenum. And then I don't see any of the lymphoid follicles in the mucosa or the submucosa.
So this is most likely a jejunum.
Because the presence of the lymphoid follicles is characteristic for the ileum, especially the terminal part of the ileum, where these lymphoid follicles, they aggregate and they can even be seen grossly on the inside of the ileum, and they are called Peyer's patches. The cell types that are present in the mucosa, the majority of cells are the tall columnar cells, the absorptive cells. Look at them here with a oval basally located nucleus, and they are called enterocytes. In between them are the goblet cells, and you can see here this hazy border which represents the microvilli, and it's called the brush border in light microscopy. In the center, there is the lamina propria extends, and you can see that there are a lot of lymphocytes in the lamina propria. There are blood vessels as well, capillaries, venules, and lymphatic vessels, and also you can see there are smooth muscle fibers. These are the muscularis propria that are extending along the long axis of the villus. In the crypts, there are cells which are called Paneth cells, especially in the base of the crypt, and you can see that they have a prominent eosinophilic granules. They secrete an enzyme which has antimicrobial properties for the defense. And also in the crypt there are stem cells. Again, they don't have any special feature, but we can say that this is a stem cell by the presence of mitotic figures. And there are neuroendocrine cells which cannot be differentiated unless we use a special stain, like immunohistochemical stain. These hormones are locally acting hormones. They regulate the gastrointestinal motility and secretion. Regarding the large intestine, the principal function of large intestine is absorption, but only of water and salt. Most of the absorption of other substances and even water has taken place in the small intestine, and also the propulsion of the content toward the exit. So you can see here that the striking feature is that because there is less absorption here, we don't need to have more increase in the surface area, so there are no villi, only crypts of Leiberkuhn or intestinal glands. And again, the same features here. There's the muscularis mucosa, then the submucosa, and then the muscularis propria with two layers of muscle fibers of different orientation. Here in a higher magnification, a simple tubular gland, no villi, and again another feature, as probably I mentioned before, there's a lot of goblet cells. The absorptive cells are the columnar cells, and the goblet cells are mucus secreting cells, because as the pieces pass along the large intestine, they become progressively dehydrated, more formed, and it becomes increasingly important to protect the lining epithelium by secreting more mucus. Of course, there are other cells again. There are stem cells. Sometimes we can see intraepithelial lymphocytes as well. This part of the large intestine we need to identify separately. It is the appendix. Note that it has a narrow lumen here. You can see part of the feces are present in the lumen. And again, there are no villi, only crypts of the intestinal glands. And the striking feature here in the mucosa and the submucosa is the presence of a lot of lymphoid follicles. And also, if you look at the crypts, they are less numerous than the crypts that are present in the remaining part of the large intestine, in the colon. This is the histology of the anorectal junction. And this is, again, another place where there's an abrupt change of epithelium. Look at the epithelium here. It is a simple columnar epithelium. The cells are columnar with oval basally located nuclei, and then all of a sudden the epithelium becomes a stratified epithelium. You can see here clearly that it is a stratified squamous epithelium. So this is at the anorectal junction. In fact, this histological anorectal junction is present between the upper part of the anal canal, this one, the upper part of the anal canal, which has the same features of the rectum. That's why we say that it is the anorectal junction, and then the lower part of the anal canal. So the anal canal has an upper part and lower part. Each one of them has a different embryological origin, and each one of them therefore is lined by a different mucosa. This mucosa is an absorptive protective mucosa, but this mucosa in the lower part of the anal canal is purely protective mucosa. Now let's deal with the liver. The liver has a dual blood supply. It is supplied by the portal vein and the hepatic artery. The portal vein, as you can see, is much bigger than the hepatic artery because most of the blood reaching the liver, about 70% of the blood reaching the liver, is by the portal vein. And the portal vein drains the blood from the GI tract to the liver. So the blood, which is full of nutrients coming from the GI tract, reaches the liver before it reaches the systemic circulation through the hepatic veins. So differentiate between a portal vein and a hepatic vein. But the blood that is brought to the liver by the portal vein is deficient in oxygen, so the oxygen requirement is provided by the hepatic artery. Both portal vein and hepatic artery, they are going to divide into several branches, smaller and smaller branches. They form a tree inside the liver, and they will flush the hepatocytes, the cells of the liver, as you can see them here. And you can see this is a branch of the hepatic artery. This is a branch of the portal vein, and together with it there is a small duct, a bile duct. The bile duct that drains the bile that is produced by the hepatocytes. The hepatocytes are arranged in a radial manner around a central vein, and this arrangement in a radial manner forms like a hexagon, as you can see here. This hexagon is called the hepatic lobule. So this is the structural unit of the liver, is hepatic lobule. Multiple of these hexagons, multiple of these hepatic lobule. The center of the hepatic lobule is a central vein, and radiating away are the hepatocytes. And at the corners of the hexagon, there are these three structures which are called the portal triad, and they consist of a branch of the portal vein, a branch of the hepatic artery, and a tributary to the bile duct. Look at it here. This is an actual section of the liver. Look at the central vein. Of course, in human, unlike in other animals, in human, the hexagonal shape of the hepatic lobule is not that clear, but still I can see here the evidence of a hexagon. Look at the corner here. These are parts of the triad, the portal triad. And as I said, that there is a central vein. And look at the hepatocytes. Multiple cells, they radiate peripherally. The spaces in between them are sinusoids, large capillaries. You can see traces of blood cells inside them as well. These are called the hepatic sinusoids. So the hepatic sinusoids, they derive their blood from the portal triad, which contains the hepatic artery and the portal vein, branch of hepatic artery and portal vein. And this blood will flush the hepatocytes and then reaches the central vein. And these central veins later on, they collect together, as you can see here, to form the hepatic veins that drain into the inferior vena cava. Look at the triad. Two sources of blood going toward the center of the hepatic lobule, two sources of blood from the portal vein and the hepatic artery, while the bile has a different direction toward the bile ducts. This is to show you the details of the portal triad, the three structures that are located at the angles of the hexagon. So this is a vein, a branch of the portal vein. Look at it. It is lined by simple squamous epithelial cells. Look at the flattened nuclei, indicating that the cell is a squamous cell. And the very thin wall, in comparison to the large lumen, because of the thin wall, it is easily compressed, and so the lumen is distorted. While if you look at the artery here, you will also find that it is lined by simple squamous epithelium, endothelium. Look at the RBCs inside. The wall is thick, look at the 2 or 3 layers of smooth muscle fibers in the wall. So the wall is thick in comparison to the small lumen, and that's why the lumen is more uniform than that of the vein. So the artery, the branch of the hepatic artery, is smaller. If you compare it with the vein, it's bigger and it is thicker in wall than that of the vein. But both of them, whether artery or vein, they are lined by endothelium, simple squamous epithelium. And this epithelium lines the whole vascular system. But if you look at the other lumen here, you will find that it is surrounded by cells that have a rounded nucleus. And the cells, they are cuboidal cells. So simple cuboidal epithelium, and this is the bile duct. So these 3 structures together, they are called the portal triad. It's not only these 3 structures that are present in the triad, but they are the most prominent. You can also here find lymphatics, but they are not easily identified. The hepatocytes, in this higher magnification, look at this hepatocyte here. This is another hepatocyte. They are large polyhedral cells. They can multiply, regenerate, and they have rounded, centrally located nucleus. Some of them are binucleated, like here, for example. 2 nuclei. This is another one, binucleate. And the cytoplasm stores a large amount of glycogen, which is washed away during tissue preparation. That's why you will find it vacuolated. Also, they are characterized by the presence of these brown granules. These are called the lipofuscin. These are what we call wear and tear pigments. They are present in variable amounts, especially with increasing age. They are not only a feature of the hepatocytes, but they are maybe present in other cells in the body, like neurons. So these are the sinusoids. They are not only fenestrated, but they are also, their basement membrane is fenestrated. And so this indicates that there is a lot of exchange, because this blood that is present in these sinusoids is full of nutrients that has been just absorbed from the intestine and has been delivered to the liver by the portal circulation. Look at this section here. You can see the sinusoids. They are red in color, while the hepatocytes, they appear as if they are empty. This slide was prepared by injecting a dye into the animal and then sacrificing it afterwards. This dye is going to reach the liver and will show the blood. Also, you will see here in this diagram that the hepatocytes, they are bounded by sinusoids on either side. So they are flushed with blood on either side. And between the hepatocyte and the sinusoid, there is a space here, which is called the perisinusoidal space around the sinusoid. Perisinusoidal space, or the space of Dease. It's a narrow space. You cannot see it clearly in light microscopy, and it drains to the lymphatic vessels and the portal tract or portal triad. Remember, I mentioned here that there are also lymphatic vessels here. These lymphatic vessels, they drain the perisinusoidal space. So substances that cannot reach the blood, they will pass in the perisinusoidal space to be drained by the lymphatic system. Another type of cells that are present in the liver are scattered among the endothelial cells that line the sinusoids. And these are the darkly stained cells here. These are called Kupffer cells. They are large cells with ovoid nuclei. They are phagocytic. They are part of the monocyte macrophage defense mechanism in the body. And they participate in the removal of senile RBCs and other particulate matter from the circulation. In this section, you can see the hepatic lobule. This is a central vein, and these are the plates of hepatocytes. In between them are the sinusoids radiating from the central vein. And here, the animal has been injected with India ink, which contains carbon particles, and then sacrificed after that. So the carbon particles, which were present in the ink, they were phagocytized by the Kupffer cells. And that's why the Kupffer cells are shown clearly here, because they contain these carbon particles. You can see the close relationship of these Kupffer cells to the sinusoids of the liver, because they are scattered among the endothelial cells. Now, before dealing with the pancreas, I would like to provide an introduction about the classification of glands. Glands are classified as exocrine glands, which secrete onto free surfaces, and that's why they need a duct. They secrete locally, so they have limited effects. For example, the salivary glands, the sweat glands, sebaceous glands, and the other type of glands are endocrine glands. They secrete hormones that reach the blood and circulate in the blood. So they don't need to have ducts. They have far-reaching effects because the secretion of the endocrine gland, like the thyroid gland, for example, is going to circulate in the blood and reach everywhere in the body. And the other type of gland is the pituitary gland, endocrine gland. And there is another gland in the body which is a mixed gland. This is the pancreas, which contains endocrine and exocrine elements. So the endocrine part of the pancreas secretes the hormones like insulin, glucagon, and some other hormones, while the exocrine part of the pancreas secretes the digestive enzymes. Also, the exocrine glands can be classified into unicellular glands, and we have seen an example of unicellular glands, the goblet cells, or multicellular glands. These glands are composed of many cells. We classify them according to the shape of the duct or to the shape of the secretory portion. So it could be a simple gland, unbranched duct, or it's compound, like a branched duct. Like, for example, the glands in the large intestine, they are simple tubular glands. Also, we divide them according to the shape of the secretory portion. So it could be like a tube, like for example, these glands are tubular glands, or it could be like an acini, a distended part. For example, as you will find the acini in the pancreas or in the sweat glands. These are acinar glands. This is to show you the parotid gland, and you can see that the gland has a capsule of connective tissue that is sending connective tissue septa, dividing the gland into lobules. And within the connective tissue, you will find that there are blood vessels, artery, vein, and also there are ducts, because these are the acini, and these acini will give rise to the duct. The distended portion give rise to a duct. This is at a higher magnification, and you can see here again the blood vessel lined by simple squamous epithelium. This is a vein. Look at the thin wall, large lumen, distorted lumen as well. If you compare it with this blood vessel, this is an artery, but these are not blood vessels, they are ducts. Look at the lining epithelium, cuboidal or tall cuboidal epithelium. These are ducts. And the duct, they don't secrete. But these structures here, these are the acini. These are the structures that secrete. And you can see that we have two types of acini. Serous acini, where the cells have a darkly stained cytoplasm, and mucus acini, like these one, which have lightly stained cytoplasm. Some of the acini, they look like this one. They have like a half moon shape here. We call them serous demilunes. So mucus acini with serous demilunes, or seromucus acini. This is a larger duct in the gland, and you can see the lining. It is stratified columnar epithelium. This is a rare type of epithelium, only present in large ducts. For example, the duct of the parotid glands and the duct of the pancreas. Here is the pancreas, and again, it's like a gland, and it has connective tissue capsule. Here, the connective tissue is stained blue. It is sending septa, dividing the gland into lobules, and within these septa, you can find that there are blood vessels and ducts. Here is again another duct. And look at the acini here, the darkly staining areas, but in between them, you can see that there are lightly staining areas, like islets in a big C. These are called the islets of Langerhans, and they represent the endocrine portion of the pancreas, while the remaining part, most of the pancreas, is an exocrine gland, which is formed of acini and duct. This is at a higher magnification. Look at the connective tissue here in the septa, and it contains a duct, and the duct here is lined by a cuboidal epithelium. And if we look closely here, this is another duct within the glandular tissue. It is a little bit lighter stained with the cuboidal epithelium, and all the other features here, or the profiles here, are of acini. This is the acinus in a diagram. So there is a central lumen in the acinus. Some of the acini, like this one, will show as if that there is a nucleus inside the center. This is what we call a centroacinar cell. It's an artifact during tissue preparation. Again, if we look at this higher magnification, we can still see that this is a duct lined by cuboidal epithelium. Look at the rounded nuclei. These are the acini of the exocrine portion of the pancreas. This is a duct here. This is a blood vessel containing RBCs. And this lighter stained area is the islet of Langerhans, which produces the hormone.